I would like to introduce you to the imaging mass cytometry, or IMC. And as you already said, it's still in the title, it's a multiparametric proteomic technology where target proteins, either in or on cells, for example, are stained with specific antibodies that are conjugated to metal isotopes instead of fluorochromes, as you would do in immunofluorescence or in flow cytometry. These metal isotopes are non-rare and non-biological and non-radioactive, so easy to work with. And then these proteins are analyzed by time of flight detection of the discrete masses of the metal tags, and not by fluorescence as you would do in flow cytometry. So that's why it's also called cytometry by time of flight. And our cyto machine can operate in two switchable modes. The first mode is the suspension mode, working mainly on the Helios part of this cyto machine, where you, like in a normal flow cytometer, introduce single particles, single cells, spray them into the machine, and then analyze them by time of flight. The other mode is the IMC mode. So you equip this module on the Hyperion module in front of the Helios. And with this module, you can insert slides and ablate small pieces of this slide tissue into the Helios machine for the analysis. So that's the two ways you can use this machine. And the outline for this talk is very simple. I would like to show you quickly and briefly how it works in general, then how you can get started and give you some help, tricks and tips and where to start and what to look at, and finish up with a practical example. So how it works. So as I said, single cells or tissue samples, also called plumes, are ejected into the Helios part. In the Helios, there is an inductively coupled plasma. So the cells are burned in this argon plasma to atoms and ions, resulting in an ion cloud that either represents a cell or represents a plume. And then these are further traveling into a vacuum where they are mass filtered and then subsequently measured by time of flight in a single detector. And subsequently, the ion counts of the different metal isotopes are then attributed back to the cell plume where they originated from. And thereby, you can get the results. So that means that the number of detectable parameters is limited at the moment by the number of available purified isotopes within the mass range of the detector. And the mass range of our detector is from 75 to 209. That means in principle, we could use 135 different markers. Currently, we can use for the SMC about 50 markers at the same time, and in the IMC about more than 40 markers at the same time. So these are the current limits, but it's steadily increasing. So with this slide, I would like to just summarize the features of the IMC. So I'm focusing just now on the IMC only, and I call it sometimes also the commercial slide because it lists all the good things about the machine. So it has a single cell resolution, so you can ablate 1 square micrometer tissue, and this gives you 1 pixel of your image, and thereby allowing an accurate and quantifiable phenotyping and cell function assessment. There is no autofluorescence when using mass cytometry compared to immunofluorescence, and that means that you can get reliable results when working with highly autofluorescent tissues like lung, gut, brain, and skin. It works well with formalin-fixed, paraffin embedded, and frozen tissue sections. And the sensitivity is comparable to that in a classic immunofluorescence system. Currently, we can roughly measure 43 targets simultaneously, and there is only minimal overlap between the detection channels that allow the very flexible marker and antibody panel setup and design. The samples from the IMC are dried, so that means you can store them over a long time and measure whenever when you have time or machine time is available. And it's a proven technology. Within the years since it has been invented, there are more than 200 peer-reviewed publications available now that can be used as a resource also for defining panels and looking for the applications. So, but on the, let's say, downside of this technology is that it's destructive technology. That means the tissues that you have ablated from the slide is not preserved, it's gone, it's burnt. And it's rather slow. So you can roughly calculate 1 square millimeter of ablation takes about 2 hours. So if you have large areas that are a lot of samples, it takes time. But and finally, the detection range for this detection range, also a lot of metals in the environment are present. So you have to be careful in terms of metals like lead, iodine, barium, and cisplatin in the sample or in your buffers in order not to contaminate your samples. So, and if you put these features into a perspective and into, let's say, special biology toolbox where CellX is providing a lot of these technologies from this toolbox, you could place IMC somewhere in the middle of technology that either have a high throughput like immunohistochemistry, or on the other end, getting or producing a lot of high data like spatial transcriptomics. And the IMC is somewhere here in the middle, allowing a good throughput and 40 plus markers simultaneously that allows it to be used in clinical and research applications, also on a larger scale, actually. So that leads to the possibility of using it for a number of applications. And this slide, of course, is a talk in itself. So I will not be able to go into details with all the applications that you can do, but in principle, you're looking mostly in tissue microenvironments by phenotyping and quantifying diverse cell types and looking at the spatial relationship in terms of immunophenotyping that can be in autoimmune disorders, in cancer, or other applications. But rather than going through literature, I would go more into the practical part and go more into how to bridge the gap between this theory and you using actually this technology. So in a normal experimental workflow, you start with your scientific hypothesis or your scientific question and say, okay, IMC can help me in getting these answers to this question. So you start your experimental planning and you usually start with a panel establishment where you put in all the markers you could think of that may be interesting to answer that question. And on the other hand, you have your tissues that needs to be processed in a way and present in a way that it gives you the answers you want. And if you have that, then can stain your slides and run it through the machine, and it will give you these images. From these, then you can do your data analysis by segmentation of single cells and so on. The critical part here for this talk and is of course and also for the experimental setup is the panel establishment, and has a number of steps, and I would like to take a little bit time and go a little bit into that. So in terms of antibody selection, well, you can get pre-labeled catalog antibodies. Mostly they are provided from the vendor of the machine itself. So this is standard biotools. And they have also some pre-designed and pre-labeled antibody kits that can be used as a start, so that you have validated antibodies to start with and don't need to start completely from scratch. And if these not good enough or not the right ones, then it's also the possibility to buy purified antibodies, sometimes also called cytometry, MaxPar ready, or BSA free, and you label them relatively easily with the metal you want. There are conjugation kits for that, and you can label about 2 to 8 antibodies in a day. And then you have 100 micrograms or maybe a little less of your antibody tagged with your label of interested. And this, of course, gives a lot of flexibility in the panel design because you put your antibody with a metal in that open channel that you have. And well, how to find these markers? Yeah, you start usually with a protolist, so everything you think of that could be interesting to address your scientific hypothesis, some nuclear markers, some structural markers, some tissue-specific markers, epithelial, stromal, immune cells, and so on. You look for the tissues that you have and what the goods and the bads about this. So normally you have either the FFPE tissue or let's say a frozen tissue section, and they have all the pros and cons in terms of availability, in terms of antibodies that are already present and so on. I will not go into detail that, but you can read up on that. And for the staining itself, it's using conventional immunofluorescence and immunohistochemical protocols, but there are also some provided from that are a little bit more imaging mass specific. So this looks relatively straightforward. And if you want to read more about on this, there are of course a lot of literature out there in the field. Some, for example, basic articles, but you can also get more specific protocol articles. The one I show here is a very recent one. What I think is quite good in terms of how to validate the antibodies and all these kind of things. And of course, the vendors, they have also a lot of information that you can use to start from. And last but not least, we have a homepage, and I also put all the information on there that I think is relevant and interesting. So you can also take this as a source. So, and I would like to finalize or with a practical example. So from our own experiments. So here I show the specifications of the machine, and I would like to point to the slide that you put into the machine, because you may think that you just put in a slide, but it needs a certain area, because there's only a certain area that the laser can reach for ablation, and this is the one here shown in violet. So the area that you want to look at, the tissues have to be placed within that area. So this is one specific thing that you need to take into consideration. And the other thing is, of course, the sample itself. The machine is recommended to be used for samples that are about up to 7 micrometers thick. 10 may also be possible. I've seen publications with that, and it also may work. But the recommendation is 3 to 4 micrometers in thickness of the sample. So let's see how it looks like. Here I show a slide stained, a human spleen tissue stained with 11 markers. And this is a picture that I took with my phone, and then you can use this image. You can also do it by a scanner or whatever you have that makes good enough quality pictures, import it into the site software, and then within the machine, there's also a photo camera that can take pictures of certain areas, and thereby you get also structural information from the slide directly in the machine, and then you can co-register your image. That can also be an H&E image or whatever you can think of that may help you to find regions of interested ROIs. And these regions of interested are then actually the ones that are ablated from the slide and analyzed. So you only take the small squared or rectangular pieces for ablation in the machine. And then that runs through the machine, it will be ablated. And when this is done, you get data out, usually a file that is called MCD file, where that includes all the information on channels, imported images, the panorama images, the ROIs, and all other information. And with this file, then you can go to a different software, depending on your pipelines, and do the analysis. Here in this case, I use the MCD viewer, and you can see that it can show you from all the different channels, so all your different markers, the intensity of the signal within the ROI, And you can also take this information to create merged images, as you see in the publications, and with these nice, beautiful overlays of different markers. So that's how it works in when you come with your stain slide into the facility, and it will be run there. That's all it needs. And then, of course, the work is not finished, because then, of course, you need to do all the data analysis. And this is not something that we provide, but of course, we also looking into that. So we decided to work us into the IMC workflow from the Bodenmiller lab that is based on Python and R. But there are also other options like IMSite, where you can use a more graphical way of trying to get the best out of your data. So that's the way it works. And the service that we provide, and this is a kind of fee for service concept. So we have some prices that for certain services, and then you can get your data acquired. And we do that for the suspension part, but also for the IMC part. However, we do not do staining or cell preparation. And with we, I mean basically me. So I probably have enough to do with the LC. And this is actually what I wanted to say. If you want to get contact with me, this is the email address. I have also open office, usually every Monday between 9 and 10 in one of the meeting boxes here in this building. So just come along and we can talk about if you're interested in this technology. And with that, I would like to thank you for your attention.